All right, our first question is, can you visually identify a cell cycle stage? Let's practice it together. First, let's look over the cell cycle and make sure we know what we're looking for. If a cell is in G1, S, or G2, it is in interphase. Interphase is where a cell spends 90% of its time. It's just doing its cell thing. So this is your cell when it's just living and being and doing its work. And then when your cell wants to divide, like if you're going to grow some more hair or replace some of those liver cells that you damaged, your cell is going to do mitosis. Mitosis has its own set of steps, which you read about: prophase, metaphase, anaphase, telophase. And then the last thing is cytokinesis. Cytokinesis is when your cell splits the cytoplasm in two. That's actually what this word means. Cyto means cell, and kinesis means movement. So your cell is moving its cytoplasm into two pieces. And it's technically not part of mitosis. It happens after mitosis. So mitosis is taking all the information in your nucleus and dividing it into two identical copies. Cytokinesis is splitting one cell's body into two pieces. So it's the cytoplasm, the mitochondria, the endoplasmic reticulum, all of the good stuff inside. So the cell cycle has 3 stages of interphase. In G1, you're going to double check that your DNA is duplicated and you're going to grow. Your DNA is going to get duplicated in S phase. Your cellular components are going to get duplicated in G1. Every time I see a G, I think grow. And then in S phase, you're going to duplicate your DNA. That's when we do that. S for synthesis phase, where you make more DNA. G2, you're going to grow a little bit more, and you're also going to double check that you're ready to do mitosis. And then your cell will divide into 2. Now, if your cell is in interphase and it's living happily, and it thinks it wants to do mitosis, it wants to divide, but there's something wrong, then your cell will go to G0. In G0, your cell cycle kind of stops and it double checks itself and makes sure it's okay. And if it's not okay, then you may trigger apoptosis and just kill this cell, program cell death. So I'm going to show you some images, and I want you to identify what phase of mitosis each image is in. Here are 5 different images. Each one of these is in one of the phases of mitosis. So take a moment, figure out which one's in prophase, anaphase, metaphase, telophase. You got it. All right, so I always look for metaphase first because I find it the easiest to identify. Metaphase is when all of your chromosomes are lined up in the center. M for middle, they're in the middle of the cell. That's metaphase. This one here is going to be telophase. You can also say telophase, totally cool, don't let anyone shame you. That's when the cell is starting to pull apart and it's going to pinch off and do cytokinesis shortly. This one is anaphase. The difference between these two is this one is showing the DNA actually pulling apart to the different poles of the cell. This one, they're already in the opposite poles. And here I have my nuclear envelope reforming. This doesn't have a nuclear envelope at all. It's already broken down. This would be a cell that is in interphase. I can tell it's in interphase and not in prophase because I can't see the chromosomes. They're not condensed yet. That means that this has to be prophase because the chromosomes are condensed, but they're not lined up on the center yet and they haven't been pulled apart. Let's do it again. Here's another set. Go ahead and identify the different stages. All right, lined up in the middle, that must be metaphase. My chromosomes are being pulled apart. That's going to be anaphase. My cell is showing its nuclear envelope coming back and it's almost ready to do cytokinesis. That means this is telophase. Here, let's start over here. Here, my chromosomes are not yet condensed. I can still see the nucleolus. Looks like my nuclear envelope is maybe almost starting to break down and my centrioles have popped up, but it's not quite ready to be called prophase yet. So this is interphase almost to prophase, and here's going to be prophase, and I know that because I can see my chromosomes have condensed and my nuclear envelope is starting to disappear. Let's try it again, but with actual cells. All right, find my favorite first. Metaphase is in the middle. All my chromosomes are lined up on the center. Then they're going to get pulled apart. That means this is anaphase. And here's telophase. They've been pulled apart enough that I'm starting to build a cell plate in between my two cells. I can still see the chromosomes, so I'm not totally back to interphase yet. But over here, I have my cell plate. I can no longer see my chromosomes and I can see a nucleolus. This means that these cells are now done and they're both in interphase, ready to just work as a cell. That leaves us with this picture and this picture. Here I can see the condensing of the chromosomes. That means that this is prophase. And here I cannot see any chromosomes. That means that this one is in interphase. So this is the start. The first step, condensing chromosomes, breaking down nuclear envelope. The next step, chromosomes in the middle. Then you pull them away, the third. Then you start to rebuild the nuclear envelope, put in a cell wall, fourth, and then last, you have two completely identical new cells. So let's take this and practice a little bit. I want you to check out this diagram. This is a diagram for causing a cell to do apoptosis. It's going to be programmed cell death. And on this diagram, I want you to identify the ligand, the receptor, an enzyme, and a tumor suppressor. All right. Hopefully you paused it and identified everything. The ligand is going to be the signal coming from your cell. In this case, these are death signals. The receptor is who responds to the signal. That would be these death receptors. So here's my ligand and the receptor. I have several enzymes in this diagram. I know it's an enzyme if it ends in ACE. So here's ACE, here's ACE, here's ACE. I have at least 3 sets of enzymes. And a tumor suppressor, we have a couple different tumor suppressors, but this is the one being shown, P53. Cool. So now we have 2 different pathways, both of which are showing us how we can lead to programmed cell death, apoptosis. Let's practice what we know. During what phase of mitosis does the nuclear envelope break down? If you said prophase or prometaphase, you're correct. During what phase of mitosis do chromosomes decondense or unwind? It's got to be telophase, because here they're still being pulled away. So here's when they're done being used and they can unwind, telophase. During what phase of mitosis do chromosomes line up in the center of the cell? My favorite phase. And that's metaphase. Metaphase for middle. Chromosomes are lined up in the center of the cell. All right. So now our second question of this lecture is, what is a mitotic index? And the answer is, a mitotic index is a way to compare how much mitosis a cell is doing. So different types of cells do mitosis at different rates. And I have a picture of an onion plant here. So this is a red onion that you would put in your salad. And here are its roots. And here is its scape, which is the green part of your plant that would grow up out of the dirt. And if you were to cut it in half, it looks like a regular onion. And the reason I'm showing you an onion is because these roots are really active mitotically. They're doing a lot of division. So we often use this in the lab to show this as an example. This is what it looks like if we were to zoom in on that root of the onion. All of these yellow boxes are the cell walls. The DNA has been stained this blue-black color. And then this is just cytoplasm around it. So we need to calculate mitotic index. And to do that, you take the number of cells in mitosis, so the number of cells that are in prophase, metaphase, anaphase, or telophase, divided by the total number of cells. So what I want you to do is count the number of cells that have visible chromosomes, not just that you can see the DNA, because if you can just see the DNA, it's in interphase. And then divide it by the total number of cells here, and that will be your mitotic index. So I'll pause it while you count them. Cool. Now your answer might vary a little bit because you could have counted this as a cell on its own. I would not count these halfies. Did you count this one? And so our total numbers are going to be different, but let's practice it a little bit together. Here's an example. A student wants to know whether or not the onion roots are growing from the base closer to the bulb or from the tip of the root. And so what she does is she takes a cross section of the root in both places, and she looks at it in the microscope and she calculates the mitotic index of both places. So you just calculated the mitotic index of the sample. This is the same image, it just got rotated. And what you do then is you can put it into, a table and compare. So areas that have more mitotic index numbers, like higher numbers, are doing more mitosis. So in this example, the student calculated mitotic index and found that the rate was higher at the root tip. That means that there is more mitosis occurring at the root tip, and so that's where most of the growth is coming from. Here's our practice question. Which of these would have the highest mitotic index number? Mitotic index is the number of cells actively dividing, divided by the total number of cells. So this would have the highest number of mitotic index, number C. Let's talk about when we actually use this in biology. And one of the examples of when we use it is trying to understand cancer cells. Cancer is uncontrolled mitosis. So if we can understand what happens in mitosis, we can understand cancer better. Here we have a table. We have the cell type. We have normal lung tissue and cancerous lung tissue, normal stomach tissue and cancerous stomach tissue, normal ovary tissue and cancerous ovary tissue. And then we have the different stages of the cell cycle. Remember, interphase is not part of mitosis. These four are part of mitosis. So what I want you to do is take a moment and calculate the mitotic index for these 5 empty boxes. All right. Did you get this? Now that we've calculated the mitotic index, we can see a trend. If you're comparing lung tissue, which one has a higher mitotic index, normal or cancerous? And the answer is cancerous. If you're comparing stomach tissue or ovary tissue, the answer is going to be the same. The cancerous tissue has a higher rate of mitotic index, and that's because cancer cells are uncontrollably dividing. Practice question. Rats were exposed to a chemical called BPA. You've probably heard about this chemical. It's what's in plastics that makes plastics nasty for health. So BPA. And then the tumor development rate was monitored in these rats. Which rats developed more tumors? Don't let this graph fake you out. Check out the labels on the axes again. So the rats that had tumor-free means less tumors. That's control rats. So even though it's higher on the graph, these had less tumors. That means that the BPA exposed rats had more tumors. Based on the data presented in this experiment, which rats would you expect to have a higher mitotic index? The rats with more tumors, the BPA exposed rats, would have a higher rate of mitotic index. What is the independent variable in this experiment? The independent variable is the thing that you change, and we change BPA exposure. Awesome. So that's the end of the lecture. Let me know if you have any questions, email me, and thank you for watching. I will talk to you soon.